I'm Deepika, working in Dr. Chris Damali's lab, Department of Biochemistry, University of Iowa. My project this summer is identifying the signal transduction pathways regulating ZO1 binding to alpha-catenin. Cells and adhesions are critical in the development and maintenance of all multicellular organisms. There are three important junctions which are present in most of the multicellular organisms, which are tight junctions, adherence junctions, and desmosomes. The most apical are the tight junctions, which form a cellular barrier to limit paracellular transport and establish apical basolateral priority. And the next come the adherence junctions, which mediate cellular adhesion by coupling to the actin cytoskeleton. The background of my project is permeability between adjacent cells is regulated by the tight junctions. However, the formation and function of the tight junctions is tightly coupled with the assembly and function of the adherence junctions. Several tight junction and adherence junctions proteins bind one another. But how do these interactions mediate junction coupling? Two very good candidates for mediating these junction coupling are ZO1 and alpha-catenin. ZO1, which is a tight junction protein, it links the tight junctions to cytoskeleton by binding to actin and then to transmembrane proteins claudin and occludin. Alpha-catenin, which is an adherence junction protein, is linked to adherence junction through beta-catenin, which in turn binds to each adherence. ZO1 is shown to bind both alpha-catenin and occludin and thereby enhancing tight junction assembly. The results from literature suggest that alpha-catenin binding to ZO1 mediates junction coupling. However, the disruption of binding exhibits impaired tight junctions and mislocalization of occludin. The factors that regulate these interactions are also still unknown. In order to validate, based on the results from the preliminary experiments in our lab, we could conclude that alpha-catenin binding to ZO1 is regulated by a ZO1 intramolecular interaction. This is the N-terminal half of ZO1. There are two regions, the GUC and U6. There are two unique regions, U5 and U6, where U6 binds to GUC, forming an intramolecular interaction. Alpha-catenin and occludin have their binding sites at the U5 and GUC domain, and so in the presence of the intramolecular interaction, alpha-catenin and occludin cannot bind to ZO1. However, from literature, we know that there is a signaling molecule, chemodulin, which can relieve this interaction, but whether this facilitates ZO1 binding to its ligands is still unknown. U6 blocks binding of ZO1 to occludin and alpha-catenin. In order to confirm this fact, we did an in vitro binding assay using his tagged ZO1 and GST tagged alpha-catenin and occludin. We could see that alpha-catenin and occludin could bind with the 516 to 806 region of ZO1. However, but they couldn't bind to the 516 to 888 regions. This indicates that there is an intramolecular interaction here which prevents the binding of alpha-catenin and occludin to ZO1. Chemodulin binds to ZO1. Using a chemodulin target database, which is an online software, we found that there were two putative binding sites for chemodulin at the SH3 region, which could potentially relieve the intramolecular interaction and enhance the binding of alpha-catenin and occludin. This is a model which suggests that in the presence of chemodulin, the interaction is relieved and alpha-catenin could bind potentially. ZO1 binding to alpha-catenin and occludin in the presence of chemodulin. We did another in vitro binding assay in the presence of chemodulin. We subjected the 806 and 888 cells to alpha-catenin and occludin in the presence of chemodulin. However, these are the results that we were able to conclude. We found that alpha-catenin and occludin bound to ZO1 in the presence and absence of chemodulin. As you can see here, there is binding in the presence and absence of chemodulin. And there were no significant difference in the levels of binding either. This indicates that chemodulin is not likely to be the signaling molecule regulating the binding of alpha-catenin and occludin to ZO1. Chemodulin inhibitor. To determine if chemodulin actually regulates this interaction between ZO1 and alpha-catenin, we studied the effects of a chemodulin inhibitor on adherence junction and tight junctions. We thought that if chemodulin regulates the coupling of tight junctions to adherence junction, then the inhibitor would interfere with the assembly of tight junctions but not adherence junction. We tested this possibility by treating MDCK cells with different concentrations of TFP, trifluoropyrazine, which is a chemodulin inhibitor. To determine whether chemodulin is necessary for coupling tight junction assembly to adherence junction, we analyzed the assembly of adherence junction and tight junctions using immunofluorescence. Adherence junctions were visualized using an antibody against each adherence, and tight junctions were visualized using an antibody against occludin. These are the results we got, and these are the different concentrations to which we subjected the cells. The tight junctions and the adherence junction assemblies were normal in the cells when treated with increasing concentration of chemodulin inhibitor TFP. So you can see that the junctions are fine in both occludin and decadherence. This indicates that chemodulin is not likely involved in the regulation of ligands binding to ZO1. Our conclusions based on this is that the intermolecular interaction in ZO1 regulates the binding of alpha-catenin and occludin. However, in contrast to what we predicted, chemodulin does not appear to regulate the binding of alpha-catenin and occludin to ZO1. Our future directions: the cells will be subjected to higher concentrations of TFP and the binding assays will be repeated to determine if chemodulin inhibitors uncouple tight junctions from adherence junction. Positive control to show chemodulin is being inhibited will be included in future experiments. The identity of the molecules that regulate the ZO1 intramolecular conformational changes will also be uncovered. Bibliography: these are the few references that I used for this project. Acknowledgements: I would first like to thank Dr. Chris Damali, working at the Department of Biochemistry, for giving me the opportunity to work in her lab this summer. And I would like to thank Dr. Alan Fanning for the ZO1 constructs. I would like to thank the Department of Biotechnology, IUSTF, SSTF, and Khorana program for funding this, and my host university, Anna University, for giving this opportunity. Thank you.